And since we've already started Chapter 2, I'm not going to go through the historical glimpse because I know we talked about it, but read through, think about those things that Pastor discovered, and we pretty much just got to this page as we were ending last time. And we're talking about the components of a typical atom, and now we know there's some subparticle components as well, but these are the major components that you guys need to know about. The neutrophils, the protons, they contribute mass to any given atom, and depending on the number of those two particles, neutrophils and protons, you're going to determine the atomic mass for that molecule based on that. The electrons have mass, but it's relatively negligible, so we usually don't consider it unless we're getting real precise. And the electrons, as you already know, are negatively charged. The protons are positively charged. So if you have the same number of protons and the same number of electrons, the charge on that particular atom is going to be neutral. Many things in nature want to be neutral, but you already know that some things don't persist as neutral, and sometimes they change in the number of electrons they might have in their cloud, or different things happen, and there's different ways to come about having different types of atomic number and structure of the atom. So the atomic number is specifically the number of protons that's in that particular atom. And so if you look down at the table that's down at the bottom here and look at the second column here, we can look at the atomic number for these. Some of these you're familiar with: hydrogen, carbon, nitrogen. Most of you have either had a chemistry class recently or you're taking one right now. And those are some easy ones that are used in many biological molecules, so you probably want to at least get familiar with those guys. But that's atomic number, and again, it's the number of protons that are in each one of those atoms. And then the atomic mass is the combination of the total number of protons and neutrons. And so we're adding again those two together in that second column. And then we can look at the number of covalent bonds that each one of those is able to form. That has to do with how many electrons are out in the outer shell or the outer cloud. If the outer cloud only has 2 electrons, or sometimes just 1, then only 1 covalent bond can be formed. But we'll look at the structure of the cloud structure a little bit here. In the last column there, it talks to you about the approximate percent of each one of those atoms down inside of the cell. And if you add those up, you're getting pretty close to 100%. So again, like I just said a minute ago, those are the major elements that you're going to expect to see down inside of components that are constructed down inside of cells. So elements consist of only one type of atom, and there's beautiful periodic tables on both sides of this room, so you can get familiar with all the elements. When you get bored, you can glance up there and start memorizing the letters and the numbers on the periodic table, and that might help you somewhere in life along the way, and then come back once you're bored with that and listen some more. So elements can't be chemically separated into simpler parts except for those protons, neutrons, and electrons, and living matter is composed of all 4 of those elements and some of the others down below, and just a few others as well. There was an interesting article that NASA put out just last year. They thought they had found an organism that actually put arsenic into DNA instead of phosphorus, but they didn't do the experiment right, so they kind of lost some credibility in that publication. They thought they'd found a brand new novel organism, and NASA's always looking for bugs out in outer space. Every time they send something to another planet, brings back soil, dirt, whatever you want to call it, from that planet, rock, and they're looking for either organisms that are alive or fossil records of organisms that might have existed in those places to get an idea of if there is life in other places. And so they went to an area of Earth that had a high concentration of arsenic in that environment, and they extracted DNA out, and they thought, based on the amount of arsenic that was still associated with the DNA, that they fed organisms arsenic as well, and they could still grow, but there were trace levels of phosphorus in there, and so the organisms were still using the phosphorus that was in that trace bit to be able to grow. So they didn't actually find something that was super novel, but it had made some news stories along the way anyway. So Electrons are arranged in shells around the nucleus. We just saw that the protons and the neutrons are down in the nucleus of an atom. And I'm going kind of fast because, like I said last time, most of you already know this stuff. And so we'll actually finish Chapter 2 today. We'll go into Chapter 3 on Tuesday. And so we've got the electrons that are arranged in that outer shell. The first shell always has 2 electrons. So if an atom only has one shell, like hydrogen does, the maximum number of electrons that can go in that shell is going to be 2. If it has more than one shell, the subsequent shell is going to have 8, and for most biological molecules, the outer shell is going to have 8. If you go to the periodic table and look at some of the other elements that are up there, you're going to find out as you get further away from the nucleus and add more and more shells on, that number changes. But in most cases, for biological type molecules that get built into macromolecules within our cell, the outer shell has 8 electrons, or the potential to have 8 valence electrons. So the inner shell is close to the nucleus. The biological molecules, basically what I was just saying, follow this, what's called the octet rule. If the outer shell is full, it's going to have 8 electrons in it. And most atoms like to have their outer shell full. And they try to have their outer shell full. They're more stable when the outer shell is full, but naturally some of them persist without that outer shell being full, and that tends to make them more reactive. It tends to make them want to react with other atoms so that they can fill that outer shell by sharing electrons. One thing you need to know about electrons, they're constantly moving. That's why we call it an electron cloud. So they're orbiting around the nucleus of the atom. Remember, the protons down in the nucleus are positively charged. The electrons are negatively charged. So you've got that electrocharge interaction that's keeping them orbiting as they're moving around the electrons. You guys probably know what makes these lights light up, right? What makes them light up? What was it? It's electricity, right? What is electricity? Did you never think about it? You know why it's not good to stick your tongue in an electrical outlet, right? Why? It'll zap you, but what does that mean? It zaps you. It'll electrocute you, but what does that mean? It almost sounds like it'll electrocute you. What does that mean? It means electrons are going to flow through your body, and they're going to do what they do. They're going to deposit energy in your body, and it's going to cause serious damage to your body. So electrons are normally moving. In these tubes, we'll come back and talk about them later, but they've got glass tubes that are filled with a gas, and when the electrons go through, they excite that gas, and that gas takes some of the energy from the electrons, and it uses that energy and converts it into light, and we call it fluorescence. It takes energy from the electrons, converts it into a different form of energy that's called light. And so that's what's going on here. And electrons are constantly moving, and so they're playing a role in the atoms of a molecule as well. And therefore, just like they're reactive in these lights, they're reactive in chemicals. They've got energy, they're moving, they can do things and change things and make reactions happen. So out in the outer shell, those electrons that are there, we call them the valence electrons. And the valence electrons are the ones that are going to be most reactive. Occasionally an electron will switch shells and another one will change places with it, but typically the outer shell is pretty stable, and those electrons that are there at a given point in time we call the valence electrons, and they're going to be involved in forming bonds. So atoms can lose, gain, or share electrons in that outer shell. And that's what we just said when we said those valence electrons are going to be involved in forming bonds. If they lose those electrons, they can form one type of bond. If they gain an electron, they can form the same kind of bond. If they share electrons, they can form a different kind of bond. And we'll look at those pretty quickly here in just a second. There are 3 main types of chemical bonds. The first is covalent bonds. Covalent bonds form when 2 atoms share at least 2 electrons. One donates one, the other donates the other, and at any given point, those 2 electrons may be in one or the other 2 clouds of that particular atom. And so that forms a covalent bond, and covalent bonds are relatively strong because those electrons are constantly changing orbits from the orbit of one atom to the orbit of another atom. That keeps those 2 atoms held together pretty strongly. And oops, I just forwarded somehow without even trying. And the next type is called ionic bonds. We'll talk about ionic bonds a little bit. Those that you're familiar with already, you've actually seen ionic bonds in real life, and you've played with ionic bonds. Some of you have even eaten ionic bonds as you were going along. In fact, all of you have, and we'll look at those in just a second. And then there's hydrogen bonds. Ionic bonds and hydrogen bonds are both charge-charge interactions. So it's kind of like when you were in grade school and your teacher gave you two magnets, and you knew that those magnets, when you tried to push them together one way, they actually tried to push each other apart. Remember that? But if you flipped one of them around, then they actually drew each other back together. They wanted to go together at that point. And if you got them close enough, you couldn't even hold them apart. They were going to go together and touch down. And again, that's an interaction that's kind of like what we're talking about here when we look at hydrogen bonds and ionic bonds. One side of the reaction is going to have a negative charge, the other is going to have a positive charge, and they're going to attract each other. And in both cases, that's the case for ionic bonds and hydrogen bonds. So these are ionic bonds, and this is the one you've eaten before. This is sodium chloride or table salt that you've eaten before. And most of you have looked down at those little particles that you sprinkle onto your meat or potatoes or vegetables, whatever you eat, and supposedly you're supposed to use less of it now because it's theoretically more healthy, but this is the guy you put on those foods to make them taste salty. And some of you have played with it. If you've made homemade ice cream, you know that you got big chunky crystals of salt when you're going to make homemade ice cream to help the water get down to, or the ice to get down to a lower temperature. And so this is an ionic bond, and basically what happens is the sodium atom donates an electron to the chloride atom, and now the chloride, because it received that electron, picks up a negative charge. Everybody good with that? And the sodium lost an electron, so now it's got a positive charge. And those two then want to interact together because one's negative, one's positive. You could do this experiment at home if you want to, and it's kind of fun to do. You might have done it when you were in grade school, but you could take a cup of water, put about 3 inches of water in the cup, and start stirring in some table salt. If you get the kind without iodine in it, it actually works a little bit better, but you can stir in the table salt until you can't get any more to dissolve into that water. Then let all of those granules that didn't dissolve float down to the bottom and pour that saturated salt solution into a glass that's clear so you can look at it. Set it in the window, and if you can do this, put a pencil across or something across and just dangle a little thread down into that water. You don't have to do that, but it tends to help a little bit. And then let it sit there for a couple weeks or maybe a month or so. Gradually the water will start to evaporate out of that glass. And as it evaporates out of the glass, now there's higher and higher concentration of sodium ions and chloride ions, and they can't stay in solution. And they have to come back together because of their charges, and they'll start to form crystals. And if you dangle that string in, you'll see those crystals forming there, and you'll get some nice, pretty, beautiful crystals there. Tell me if you play with that. It's fun to do, and you'll get an idea of how ionic bonds form. Water is also ionic, right? You know it's got negative charge on one side, positive charge on another. We'll look at it in a minute. So when there's enough water, those ions can stay separated from each other because the negatively charged chloride wants to go to interact with the hydrogens, and the positively charged sodium wants to interact with the oxygen, so the molecules stay separated. We call it dissolved. But as the water volume decreases, they can no longer stay separated from each other anymore, and they begin to form those crystals again. There's another way to look at it. The crystals form like this, and so we've got the sodium here related to the chloride, and they pack in as tightly as possible. You may have done this before. If you get one of those salt crystals that you would put in, not the ones that you put into your water softener, because those really aren't crystals. They're just compressed sodium chloride, but get some of those crystals when next time you make homemade ice cream. Take that crystal, get a knife, and just very gently tap the crystal. Don't chop all the way through. Just tap it. And what you'll see is the crystal will fracture. And both sides of that fracture line will be very smooth and very clean, because what you did was you just broke this crystal between the sodium and the chloride, and it makes a nice smooth fracture line. Occasionally you'll get a little jagged area. There was some impurity in there that wasn't allowing the elements to come together as tightly as possible. You should play with that. I think you'll get excited about chemistry if you actually do some things like that. So when atoms have a positive charge, we call them cations, and when they have a negative charge, we call them anions. And cations and anions tend to react together. They form ionic bonds, again, because of those positive and negative charges. ionic bonds are relatively weak by themselves. But if you take one of those salt crystals that you use to make ice cream, it's not real pure salt, so don't necessarily do this, and if you get sick, it's not my fault. But you can do it this way. I was going to tell you to bite on it, but don't do that. Just put the crystal on the table and push on it with your finger. And you'll find that because of all those interactions, it's pretty hard to break that crystal. If it's a real crystal, you probably won't be able to crush it with the force that you can put down on it with your finger or your thumb. But if you take something that's a little bit more solid, like the side of a spoon or a handle of a spoon or a knife or something like that, and push down, you can break those bonds with the force that you can exert there. So play with that and you'll get familiar with ionic bonds. Then there's covalent bonds. Covalent bonds share electrons. So in this case, we've got hydrogen, and if we look at carbon, carbon's got 4 valence electrons. Naturally in nature, carbon's got 4 valence electrons when it's in elemental form. And those valence electrons, remember that outer cloud wants to have 8 electrons. So you can get 4 hydrogens that each have 1 valence electron reacting with carbon, and they'll go together and they'll make this molecule called methane. What do you guys know about methane? It smells a little bit. What else do you know?
Planets are made of it.
What?
Planets are made of it.
Planets tend to have some in their atmosphere. We can get more if we have cattle producing it, or humans. I won't tell you about some experiments that some guys in my dorm when I was in college did. But yeah, so what else do you know about methane? It's organic, and I was looking for the flammable part. There's lots of energy stored in those bonds. And if we break those bonds, the energy is released, and therefore it's flammable. We can get it to go up into flames. So there's lots of ways to represent molecules, and you've seen all of these. This is the most basic form showing you the actual electrons. Sometimes we represent with single lines, and if you see the single lines in between those elements, that means that and it's a covalent bond, that means there's 2 electrons being represented by that single line. Sometimes we see this ball and stick model, sometimes we see this space filling model, and then we can obviously just represent any atom based on its chemical formula. For methane, the chemical formula is CH4. There's 1 carbon, 4 hydrogens, all covalently linked together in that molecule. They come together because, again, the carbon and the hydrogens want to have their valence electron filled. Covalent bonds are strong. Covalent bonds are harder to break. 1 single covalent bond is stronger than many, many hydrogen bonds, or many, many hydrogen bonds. And so at biological temperatures, it's difficult to break them. To get those bonds to break in methane, you have to add some energy source. Usually you have to add a little bit of heat in the form of fire to get those bonds to break. And then once the fire is there and you begin to get bonds breaking, you get more energy, and that can continue the reaction going. So if you think about biological molecules, the molecules themselves are typically held together by covalent bonds, but sometimes we need to break them, right? In anatomy physiology, you learned about glycolysis. That's taking glucose down to pyruvic acid. That molecule has been broken up a whole bunch of different ways in the process of glycolysis. It's got to happen, but most cells can't live in the presence of fire. Very few can, but those biological reactions, breaking covalent bonds, still have to occur. So we need something to help with that, and that's enzymes that are going to help with that process of breaking those covalent bonds. Enzymes are able to catalyze the same reactions that we can do chemically, but typically require much less energy to get the reaction started. We'll come back and we'll talk about those guys a little bit more, enzymes a little bit more as we go on through. So covalent bonds can have a couple of different properties. You know that they share electrons. That's what the definition of a covalent bond is. But sometimes, if you've got a friend, a significant other, or a spouse, you know sometimes we don't all share equally. In the mornings, don't get mad at me if you're listening to this, honey, but in the morning sometimes I have to go look for the toothpaste. I usually leave it out on the counter so she can find it. She almost always puts it away. She doesn't share the same way that I do, and some atoms are the same with electrons. Some will take those electrons and they'll hold them tight and pull them away from the other one. That's my wife, and with the toothpaste, not with the electrons. And that means that that one has a net negative charge, right? The other one's going to have a net positive charge because the electron's being pulled by the other atom away. And so you can see that this is water right here, H2O, and it's water, and the oxygen tends to like to pull those electrons toward it. Therefore, it tends to have a net negative charge, and we write it as a delta negative. It's not a full negative like it had an extra electron, but it's a partial negative charge. And the hydrogens, because the electrons are being pulled away from them toward the oxygen, tend to have a partial positive charge. And that makes molecules polar. That makes them have polarity. Just like those magnets that we talked about a little bit ago, they're also polar because of what's going on inside of the molecules down inside. And that figure there's just trying to show you that the oxygen has more affinity for the electrons, so you're getting more electron density when you go toward the oxygen side. If molecules share the electrons equally, meaning at any given time the electrons are equally in one cloud or the other, then those bonds are called nonpolar bonds. So if my wife and I both just left the toothpaste on the counter, we'd be very nonpolar, and yet we seem to be polar, at least over toothpaste. So that gives you some ideas of how chemicals work. And one atom typically is going to be more electronegative than the other in a polar type of bond situation. And polar bonds are very important for biological molecules. They help things like proteins stick together. They help them fold correctly, and they help them stay folded correctly. Ionic bonds contribute to that too, and covalent bonds do as well a bit. But polar bonds are very important for many things. And polar bonds can result in the formation of hydrogen bonds. Let me just check background real quick to make sure what you guys know. From your other biology classes, including anatomy, physiology, or what other ones do you have, what large macromolecules that are down inside of the cell are held together by hydrogen bonds? Proteins are held together where at? How do hydrogen bonds hold proteins together? I want to make sure we know. Are the amino acids linked together by hydrogen bonds? Those are covalent bonds, right? They're covalent bonds between each one of the amino acids that make that polypeptide chain, but then what happens? Secondary structure, the protein starts folding down on itself, and the side chains over here may have a negative charge, and another side chain over here have a positive charge because of a polar bond, and we can begin to get a hydrogen bond forming that starts to lock that molecule in the folded position that it needs to be in. So we begin to see that. You can see that here. We've got two proteins interacting in this figure here, and on some places on each of those molecules there are positive charges, and in other places there are negative charges, and those charges begin to draw those molecules together, and the more of those charges, the more of those hydrogen bonds or polar interactions that are happening, the tighter and stronger those molecules will be held together. So as you think about that, if you had a mutation in a gene, a change in a gene that coded for this protein, and it changed that amino acid that has a positive charge to a negative charge, what do you think these proteins would do? Would they interact as nicely together as they would like they originally were? They wouldn't, right? Because this area right here would begin to repel each other. It wouldn't let them come together like they had previously. It might completely interfere with the function of those particular proteins. So if you get numerous hydrogen bonds, the interactions become stronger and stronger, and obviously it's important for a lot of different things, including molecular recognition. These bonds are relatively short-lived. You know, when you took those magnets and you put them together and they stuck together, you could exert enough energy to break those interactions, and in the cell these types of bonds can relatively easily be broken as well. There's a lot of things you can do. Remember when we talked about the sodium and chloride in water and how the sodium ions and the chloride ions would go and interact with the the hydrogen and oxygen. If you add salt in here in between these molecules, in other words, if you put it in the solution and it gets down here, it'll start interacting and breaking some of those interactions so that the strength is weakened between those molecules. So one way to think about this is, have you guys looked down closely at Velcro? Do you guys know how Velcro was discovered? Some guy took microbiology and then he went hiking out in the forest. And in microbiology, he learned to think a little bit. And then he went out hiking in the forest. I don't know about the microbiology part, but he went hiking in the forest and he found when he got done hiking that he had things stuck in his socks. And he actually went and looked at those things that were stuck in his socks and he pulled them off and he looked at them under a microscope and said, they have little hooks down on there. And he thought, that's kind of neat. They stick pretty strongly. What if we put hooks on something and loops on another side? Those will interact pretty strongly together. And that's how he figured out to invent Velcro. And he's a multimillionaire now because pretty much everybody that has ever made Velcro gets to pay him some money for that. So he just was observant, asked questions, figured out the answers, and said, how can we use that information? And that's really what I'm hoping all of you are going to do as you go through this class. You're going to learn to make observations, ask questions, and figure out how to use those. Someday you're going to cure cancer, figure out how to deal with some of the parasites that we don't know how to deal with yet, or a whole bunch of things. But as these negative and positive bonds come together, if you just had one hook and one loop, it would be pretty easy to break them apart. But the more of those hooks, the more of those loops that you have linked together, the stronger those interactions are. And so that's the case in molecular interactions as well. Somebody asked me about moles the other day, and moles are always a fun thing. There's actually an annual moles day to celebrate this number and the person that figured out this number. But basically, this number, 6.022, and it goes on and on and on, times 10 to the 23 particles is called 1 mole. And so you can have 1 mole of anything. You could have, if you were a really, really, really big university, you could have a mole of students. There aren't probably a mole of students in the world active right now, but you could possibly have a mole of students. You could have easily a mole of sodium chloride, or you could have a mole of sucrose, your table sugar, or you could have a mole of Volkswagen Beetles. Or you could have a mole of Cadillacs or Learjets or whatever you want a mole of. Just get that much money. If you get a mole of Learjets, give me one, please. But you could have a mole of just about anything. It means you have that many, 6.022 times 10 to the 23 of those things. And you figured out as I was going through all of those strange examples that a mole of any given substance, whether it's a Learjet or a mole of sodium chloride, they have different masses, right? And so it's the same with chemicals. If you get a mole of one chemical, it's going to have a different mass than a mole of another chemical. And here are just a couple of examples. If we look back at sodium chloride, that table salt that we just talked about, a mole of sodium chloride weighs 58.4 grams. But if you look at one of its cousins, if you look at the periodic table, they're not too far apart. The potassium is just one step down below the sodium. But if you make a mole of it, it weighs 74.55 grams. So it's got very significant difference in mass in 1 mole. Again, it's the same exact number of atoms, but the mass is different. And the reason for that is because of the difference in the mass of sodium and potassium. You can look at the periodic table when you get bored and do the math yourself. So, like I said, you can have a mole of anything. And if it's a chemical, we could take a mole, we could take 1 mole of a chemical like sodium chloride, put it in 1 liter of water, and we would have made a 1 molar solution of sodium chloride. If we took half a mole, if we took, let me see if I can do the math quick, 29.2 grams of sodium chloride and put it into a liter of water, then we'd have a 0.5 molar solution, and so on down. You can make a 2 molar or a 5 molar, as much as you can get in there. When you guys are making that saturated solution, it's going to be approaching 5-6 molar sodium chloride when you get down into that saturated state. So, just for an example, we talked through it, but 58.4 grams dissolved in 1 liter of water would make a 1 molar solution. So this is water again. You can see it. We just saw it represented this way previously. What do you guys know about water? You need to drink it. It can be cold. It can be hot too, right? So it's a universal solvent, and that property is because of the polarity that we just talked about. But let me ask you this. It's supposed to precipitate tonight or tomorrow, right? And if it was precipitating in the middle of the summer, you might even be brave enough to venture out and go walking in the rain with the one you love, right? You might not have a problem with that. You'd go out, you'd sing, you'd dance, you might swing around a lamp pole. You've seen the videos and the movies, and you might just do that, right? And you might be happy about it. Everything's good, right? Mm-hmm. But if it decides to precipitate a lot tomorrow and that same water becomes solid and makes spheres of water, how likely is it you're going to go dancing in the water coming down from the sky with the one you love? Yeah, if you do, you're the one you love is probably thinking, did he or she just take out an insurance policy on me? Why are they trying to get me out there? What's the difference between those two types of water? One is liquid, one is frozen solid. But what's the real difference between them? Temperature makes the difference, but what's the real difference between them? Let me ask you this. Why are you okay getting hit with water but not getting hit with those frozen water molecules, or liquid water, but not the frozen water molecules that come down and we call it hail? Why don't you like hail? It might hurt. Why might it hurt? It's hard. Why is it hard? The molecules are frozen. How does that make it hard? The hydrogen bonds are stuck. They're not moving. That's part of it. But what else about the hydrogen bonds? There are more of them. In liquid water, the hydrogen bonds are always breaking, forming, breaking, forming, breaking, forming, breaking, forming. In solid water, they're constantly fixed together, and there's roughly 4 bonds per molecule, and that's a lot of energy holding that together. And so that's why it hurts when hail hits your head. Or 2 semesters ago, there was a gigantic hailstorm in Dallas. Some of you might have been up there. One of my former students was there, and he posted this picture on Facebook, and I had it in some old lecture notes. It's not in this set, but his car got totally pummeled. The top was bashed in, the back window was bashed in, and it was just destroyed. He told me later they decided to rebuild it, and I can't imagine how they did. But in that picture, the little corner where his UMHB parking sticker was, I said, well, at least you still have your parking sticker. It was still there, and it wasn't damaged, but that was about all that was left of the back window that he took the picture of. So there, you got to find the silver in the clouds, right? And so anyway, hail can do a lot of damage, and it's because of all those hydrogen bonds that are holding the molecules together. If we take this water, somebody said it's cold, and it could be cold, and we can simply remove the energy from this liquid water, and it becomes cold when we do that, and it'll transition into this phase, because as we remove the energy, those molecules slow down and slow down and slow down and begin to align and form that crystalline state. Go to your freezer tonight, grab an ice cube, get a small knife, just tap it on the cabinet. Don't do it on your hand. I don't want you to cut yourself. Set it on the cabinet. Just tap it, and you'll see that it too fractures, and it'll fracture right along one of these lines where you have the hydrogen bonds, and you can break hydrogen bonds, and you'll see how smooth those molecules are when you do that. So you can remove the energy from this water, and it'll go that way. What happens if you add energy to this water that's liquid? It boils. And when a boiling happens, some of these atoms you see don't have hydrogen bonds, and they start to jump out. And that's what boiling is. The water molecules go into a gas phase. They're no longer associated with each other and being held together by those hydrogen bonds. But if you were doing it in a pan and you put a glass lid over it, you would see that pretty soon you got water droplets on that glass lid again. When they're getting there, the energy is being removed by the glass and the cool air, and now they're going back and starting to make hydrogen bonds again. So that's some properties of water, and those are basic properties. Water makes up 70%, sometimes even more, of living organisms by weight. It's a polar molecule we already talked about, and it has hydrogen bonds, which explains those properties of ice crystals and liquid and gas phase as well. And we've already talked about how you can take polar molecules, put them into a polar solvent, which is water in one example, and those polar molecules will dissolve down. And this part of the figure is showing you exactly what happens there as those ions become surrounded by the charged parts of the water molecule. We call that dissolving. So the compounds are hydrophilic, That means they want to be in association with water. Literally, it means they love water. And then other molecules that are nonpolar are typically hydrophobic. They don't want to be in water. Literally, it means they're afraid of water. You've played with some of those too. When you were going to make pasta the other day, you put water in the pan, you started heating up the water, you might have put some salt down, and that was that ionic reaction. You saw it dissolve pretty quickly. And then you took one more chemical and you poured it into the water, and that was probably some vegetable oil. And when you poured the water in on top, the oil in on top, you saw it go down into the water, and then it floated back up to the top, and it kind of made a layer on top of the water. Remember that? You've seen that. That's because oils are nonpolar, and they're hydrophobic because of that. They don't want to be with the water. So even though they went down into the water because of the force of them entering, they were surrounding themselves and keeping themselves separated from the water. And as quickly as they could, because of their density, they floated back up, and they made a layer on the top that separated them from the water. Those types of nonpolar molecules are hydrophobic. They don't want to be associated. They don't freeze well. If you put chemicals into water, it will freeze at a lower temperature. In other words, you'll have to take more energy out. So you guys have done that. You didn't know you were doing it, but when you made that homemade ice cream, if you just put ice in and packed ice around the homemade ice cream, you could churn it all day long and it wouldn't freeze, because the ice isn't cold enough to freeze the things that are in the container, because there you've got stuff that's mixed into the water, and that stuff that's mixed into the water lowered the freezing temperature. So you have to do something to make the ice get colder. You have to put salt on it, and when you put salt on it, it makes the ice actually get colder than the normal freezing temperature of water, and that lowers the temperature down to a point where the ice cream can freeze. And that's basically what that line is saying in that statement, that water with things dissolved in it is going to need a lower temperature to be able to freeze. All right. pH is fun, and we can talk about pH of aquatic or aqueous solutions. pH basically is the measurement of the concentration of free protons or hydrogen ions down inside of a solution. So you know some things are acidic, some things are alkaline or basic, and if you look at the scale, you're familiar with some of these guys. My favorite one is this lye. If you watch old TV shows and ever watch the Beverly Hillbillies, Granny always drove Mrs. Drysdale crazy when she went out to the cement pond. You remember that? She had this big kettle out of the cement pond, and she would take animal lard, and she would take the animal lard and she would throw some lye in with the animal lard, and she would make what we call lye soap. She was putting something that was very basic in the animal lard, and it would break some covalent bonds in the animal lard, and it stinks to high heavens, but when it comes out, it congeals and it makes something that functions as soap. And she was making that. Usually you're not going to use it on your body, but to use to clean clothes, because it's pretty caustic for our skin. But that's what she was doing out at the cement pond. If you go, a lot of you ladies have long hair, so I imagine you clogged a drain somewhere along the way in your life and your dad had to get some drain cleaner. That's usually potassium hydroxide, and it's got a pretty high pH. It's pretty alkaline, and it goes in and it breaks all those covalent bonds in the proteins that are in your hair, makes them break apart, turn jelly-like, and then you can wash them out of the drain. If you go to the other side, we've got a couple things down here that you're familiar with. You probably know it's not a good idea to explode the batteries that are down inside of your car. It's not too hard to do if you're not careful, if you short them out. But that's sulfuric acid. It's usually about 1 normal sulfuric acid. And we say it's the concentrated as we can get, even though we can go a little bit more. We use it for the scale to be a pH of 0. Okay? And stomach acid is about 1.3, just for reference. And some of the other things that you guys like, lemon juice is right up there around 2.2. Coca-Cola is about 2.7. When I was your age, I used to drink a lot of Coke. My dad said, Greg, you're going to rot out your stomach drinking so much Coke. You're adding all that acid into it. And I said, Dad, look, I was pretty smart. Look, stomach acid is more acidic than Coke. It's not going to hurt me. But in the end, he was right. I ended up getting some ulcers, so I gave up Coke, and they pretty much went away. So dads are pretty smart. Moms are too. How many of you have ever gone to Starbucks and had I know you've been there, and had a big cup of coffee, and 20, 30 minutes later, you started feeling some heartburn or acid reflux? Do you guys have that? It usually gets me if I have something that's been cooked and it's got grease in it. And that's because coffee has a good bit of acid as well. And then when you put the food on top of it, your stomach starts making more acid, and so it causes the pH of your stomach to drop way down. If we look at unpolluted rainwater, it's getting close to neutral. It's still below 6, and the most pure water that we're able to get very effectively has a pH of around 6.2, 6.3, 6.4. Theoretically, if it was perfectly pure water, it would be a neutral pH. The concentration of hydrogen and hydroxyl, that's the other molecule that's going to be there, would be exactly the same. What's water made of? Hydrogen and a hydroxyl. A hydrogen looks like this, and a hydroxyl looks like this. They come together and they form H2O. So in ultra pure, the highest purity water, if we could get there, it should be right around neutral. In this building, it's about 6.4, 6.5. So we're getting close to there in this building. And then your blood's always maintained just above neutral. That's one of the tests that if a person's not well, the doctor will immediately do. They'll look at the pH of the blood, because if the pH is going up or down, that means there's something wrong with some organ in the body or some infectious process that's causing the protons to be released or hydroxyl groups to be released. And then seawater is right around 8.2 or so. That's why a lot of evolutionists think that life on Earth started in the seawater, because that pH is pretty hospitable for organisms to be able to grow. When you wash your clothes, you use some kind of laundry detergent, they're usually around a pH of 10. Again, the alkaline nature of laundry detergent breaks those covalent bonds in the molecules that are stuck to your clothes, and it helps them to be able to release those things that cause stains on clothes. And so those are some things that you're familiar with. pH is just a relative measurement of the proton or hydrogen concentration in volume. So let's see, we talked about this stuff already. So when we go up the scale like this, up this pH scale, it actually means the concentration of protons is decreasing. If we go down the scale this way, it means the concentration of protons is increasing. And you can see that in the scale that's here, although it's a little bit cryptic. If we look right down here, it says 10 to the 0. Do you guys know what 10 to the 0? How would you translate that into some number that's easy to say? 1, right? 10 to the 0 is 1. And so in science, we always use 1 to represent as much as you can get. That's why on your microscopes, on the switch, when you turn it on, the side you push has a 1 on it. That means give it all the power that can come out of that outlet. Open it up and let all of it come through. That's what 1 means. The other side of that switch on your scopes has a 0. And look what you're doing when you go to the other side of this. You're at 10 to the minus 14. Do you guys know how to translate 10 to the minus 14? It looks something like this. If I can get the screen to erase. 0.02, 3, 4, 5, 6, 7, 8, 10, 11, 12, 13, 14. It looks like that. I didn't quite have enough space. That's getting pretty darn close to 0. Would everybody agree? Yes. Yeah. So that's what the other side of the scale is when you go back that way. And now I got to get out of that and go back here. And so that's how this scale works. At the top, you're approaching 0 concentration of protons in solution. That means there's going to be a lot of hydroxyls there. The concentration of hydroxyls is also always inversely proportional. When we get to the neutral point, again, I said it, but I want to make sure you got it. At neutrality, the concentration of protons and hydroxyls are the same. That's why I said if we had ultra-pure water with nothing else in the water but water, the concentration would be exactly the same. Okay. So again, I know I'm going really quick, and some of this may not sink in, but think about it and then come ask me during office hours questions that you have about this chapter. So we start using those atoms to build elements and molecules in cells. And basically, if you take all the water out of cells, approximately 1% of the cell is going to be non-water stuff. And it's all made out of those elements that we just looked at. And we talked about how molecules are held together, one by covalent bonds, but then by hydrogen and ionic bonds. And these types of structures on those biologically important molecules are going to hold them together. These guys cause proteins to fold as they would. These guys make other chemicals want to interact with each other. And I would just get these. In carbohydrates, you've got aldehydes that hold the carbohydrates and tend to be reactive in those carbohydrates and oftentimes on side chains of carbohydrates. And in a little bit, in Chapter 3, we're going to start looking at biological molecules that are important to bacteria and other microorganisms. So I might just say aldehyde, and I'm going to think that you already know what it is because now you've seen it. Amino groups. Where have you seen amino groups? On amino acids. That's how they got their name. On one side of the amino acid, they have that NH2 that's reactive, and that's part of what links it together. Carboxyl groups. Where have you seen those? Same place, right? The other side of the amino acid has a carboxyl group, and the amino group on one amino acid and the carboxyl group on another can interact together and form that covalent bond. Hydroxyls, they tend to have a net negative charge. By the way, all those oxygens out there, tend to have a negative charge, and that makes them polar. Remember, oxygen wants to pull electrons toward it, and that helps it contribute in those ionic bonds that we've been talking about. Keto group is another one. We just talked about methane. This is a methyl group right here. Over here you'd have some R group attached to it with that methyl group, and those have very important biological function down inside of the cell as well. And then phosphate. You've seen that before. Where have you seen phosphate? What kind of biological molecules have phosphate? ATP does, DNA does. There's one other important one. GTP does. It's kind of like ATP. Phospholipids do. Good. So there's still one more I'm thinking of. RNA. Beautiful. You got most of them that have phosphate. So you guys know this stuff pretty well. Hopefully if you do a quick review of the chapter, you'll get most of the significant parts. And then there's sulfhydryl groups. This is one that students usually don't remember from anatomy, physiology, but it's really important for protein structure. Think about if you're that organism that lives out in water that's at boiling temperatures. And that's where you grow and divide. Remember I told you, if you put your hand under the faucet at 65 degrees and leave it there for a few seconds, it's going to make your skin sore, it's going to make your skin hurt. Some of your proteins, even at that temperature, are going to denature and fall apart. So where do you think sulfhydryls can be used? Do you remember? What's that? In the ocean? Maybe oceans usually, except for at the floor, don't get that hot. But they're used pretty much every place. They're using cysteine. Cysteine is amino acid. Remember that? It's one of the two amino acids that has a sulfhydryl group. And you can get disulfide bonds when those two covalently bond together. So one cysteine here, remember we talked about hydrogen bonds, and ionic bonds can hold proteins folded together. But now if we have a cysteine here and another one over here, and they come together and they cross-link and form a covalent bond, even boiling is not enough temperature to break that covalent bond. And so for those organisms that live at very high temperatures, they have to put a lot of disulfide bonds into their proteins to keep them from denaturing at that temperature and to keep them active and functional at that temperature. So these are, again, the major and minor elements that are important. Some of these guys are all important for those biological molecules. Some enzymes need calcium, magnesium, and iron to be able to function correctly, and we'll talk about potassium in that case as well. We'll talk about those down the road. Those functional groups are very important for structure. Somebody said ATP just a minute ago. You can see the three beautiful phosphate groups on this ATP, and they're out over here, and you already know that this bond and this bond have high energy. What that means is if you break that bond in a way that you have the ability to capture the energy that's released when it's broken, then you can use that energy to do something else. And it has more energy than this bond or these bonds over here, even though they have some degree of energy. So you know ATP, it's kind of like the silver dollar of the cell. It's what drives most chemical reactions. You mentioned GTP, it can drive some other reactions. And so ATP basically provides the energy, and the energy it provides, again, is through controlled breaking of usually this bond first, and then sometimes this bond to release the energy out and be used for something else. Those phosphates have a negative charge, and so they tend to repel other things. You can see the oxygen surrounding the phosphates. That gives them a negative charge. Bonds are inherently unstable, easily broken. And when they're broken, they release a lot of energy, whether or not you have enzymes that can grab that energy. And the little tilde there indicates that you have high energy. When you break this bond, then you make ADP and you release an inorganic phosphate into the environment. If you were then to subsequently break this bond, you would release another inorganic phosphate, and then you would make AMP, adenosine monophosphate, right? Everybody good with that? So in the cell, you know, we have lots of macromolecules. You just named about 5 different classes as we were talking just a minute ago. And we can build those macromolecules. In fact, the cell does that. And sometimes when the cell builds them, it does a dehydration synthesis. In other words, a hydrogen from one of the 2 molecules and a hydroxyl from the other is actually kicked out during the reaction process. And when it's kicked out, water is released into the environment, and then a covalent bond is formed in the place where that hydroxyl and hydrogen were. And that's called a dehydration synthesis. Other times, hydration reactions happen. In that case, water's involved again, but in this case, water's added back in where some covalent bond is, and you see the 2 components of water being added back in, and now you get those one individual molecule being broken into 2 components because of that hydrolysis reaction. So there are 4 major classes of macromolecules: proteins, carbohydrates, nucleic acids, and lipids. You guys just named all of those. Those are all polymers. They're formed via this dehydration synthesis type reaction. Every one of those involves a dehydration synthesis reaction. So every one of those, as subunits are being added, involves a reaction that looks like this. Okay, so just keep that in mind as we're going through and as you're thinking about this chapter. And as they do that, water is always yielded from that reaction. Sometimes it's called biological water, sometimes it's called molecular water. It's because water is being released from those molecules that are being formed. And then, like I said, you can do that hydrolysis reaction to break those same types of molecules back together. Hydrolysis reverses the reaction of dehydration synthesis, and it yields water as that's happening. All right, so proteins are important. They're at least half of the dry weight of most cells. You know they have a lot of different functions. They can catalyze reaction. They can transport molecules into the cell. They can even move cells around, and they provide a framework or structure for the cells. And many times proteins are also involved in sensing what's going on out in the environment. Today I'm going to go try to put the video up for you guys to see of this cute little bacterium sensing that there's a neutrophil. The neutrophil senses the bacterium and the chase begins. The neutrophil starts chasing. The bacteria says, oh, he's getting closer. Even though they don't have eyes, they can tell that they're getting closer to each other. The bacteria tries to run away because he knows there's something bad's coming. And the neutrophil figures out which direction he's going. And after about 15-30 seconds, finally the neutrophil chases him around all over the field and figures out. And they're using these types of proteins to sense where the other one is. So the bacteria says, okay, he's coming this way. If I cut off this direction, maybe he won't see me. But the neutrophil pretty soon figures it out and follows him down the trail that he left of chemicals as he was moving around. Proteins are also involved in turning on and turning off different genes down inside of the cell, so that's obviously important. And proteins are made up of these molecules that are called amino acids. You guys just described these to me just a minute ago. Remember, we've got the amino group on one side, and we've got the carboxyl group on the other side, and they can be linked together by the next amino group coming in this way. The other thing that relates back to the historical glimpse in this chapter is this part. You can take those same things. You can take carbon and amino group and a carboxyl group. The center carbon, by the way, is called the alpha carbon. And you can put all 4 of those things, including the side chain that's kind of buried there, onto that center carbon, and you can make amino acids in 2 different ways. You can glue them together in different ways. And one way you're going to get an amino acid that's called the L stereoisomer of the amino acid, and in the other you're going to get what's called the D stereoisomer. And it's kind of like when you do that selfie in the mirror. When you do the selfie, you're never exactly the same because you're flipped around backward when you take it from the mirror, and it's kind of just like that. They're mirror images of each other, and if you look at those guys and flip them over, you'll see that they too indeed are mirror images. The X that they put here is here on this side. It's over on the opposite side. Can't get to draw over there. And this molecule's here, here, and so they're glued together. All the components are exactly the same. The molecular mass is the same as well, but functionally they're different. It's that experiment that Pasteur did in the historical glimpse, where he tasted those two different stereoisomers, and he saw that they tasted different, and that's because they interact with the receptors in the tongue differently. They do other things biologically different as well. So you guys, I know in anatomy and physiology, memorize all 20 of these essential amino acids. I'm positive you did, even though you're shaking your head no. I know you looked at the page once. If you keep shaking your head no, I'm going to tell the professors to let you take that course over. So just stop right there. But these guys are important. These are the building blocks for proteins. These guys are called essential amino acids because you need these to make proteins. You have to have most all of these in your diet to be able to make the muscle tissue and other proteins that are in your body. Your body can't synthesize most of these amino acids. But bacteria, remember, they're biologically diverse. E. coli, for example, it can make all 20 of these amino acids. You don't have to give E. coli, if it's a normal wild type, a normal E. coli, any amino acids. Give it glucose, give it all those major and minor elements that we just talked about. It'll be able to make all of the amino acids. It can make all the nucleotide components to make RNA and DNA. It can synthesize all the fatty acids that it needs to make lipids. But we call them essential amino acids because you guys need them to make your proteins. Well, I do too. And so we can look at these guys closely, and if you look closely, I'm not expecting you to know all the names, but those of you that are biology majors, probably the next class you take, you're going to need to. And so I would start to get familiar with these names. There's a few I do want you to know. But if we take this group, this group right here, and look at these guys, they all get called nonpolar. What does nonpolar mean? They're hydrophobic, and they're hydrophobic because there's no charge on them. And that's literally what nonpolar means, that no part of the molecule has any net negative or positive charge except for those amino and carboxyl groups. So we're talking about the side chains now, those R groups, and that's the only difference. If you look at each one of these, each one has an alpha carbon, each one has a carboxyl group on one side, and each one has a nitrogen group. I'm running out of space, but each one has a nitrogen amino group on the other side. The only difference is what's added to that alpha carbon. So we talk about 20 essential amino acids, but you could synthesize an infinite number of amino acids. How would you do that? Just add a different R group. Make some different molecule, stick it on that base subunit of an amino acid, you've got a brand new amino acid. These just happen to be the ones that our ribosomes and tRNAs are trained to use to build proteins. So these guys are nonpolar. These guys are called polar, and they're also called hydrophilic. They're also called uncharged. And now you're thinking, wait a minute, how can they be polar and hydrophilic and uncharged? And what that means is if you look down, there's no negative or positive charge on the side chain. But there is that delta positive or negative charge that we talked about on those side chains, because look right here, there's a hydroxyl group. Remember, oxygen always pulls the electrons toward it, so there's a little bit of a net negative charge there. Over here as well, and over here and here and here, you've got oxygen hanging out there. And again, that oxygen is going to pull the electrons toward it. It's going to have a tendency to have a net negative charge. And so even though it's not got a full charge, therefore we can call it uncharged, it is polar. That's what polar means, right? And so those guys tend to be reactive. Those guys tend to want to associate with water. So think back to the oil and the salt analogy I just gave you a minute ago. If you put these guys, any of those guys, into a protein, where do you think they're going to want to be in a protein that's living down inside of the cytoplasm? They're hydrophobic. And they're in a long polypeptide chain. Which part, as it starts to fold together, where do you think it's going to want to be? It's going to want to be folded down inside, because remember, the whole cytoplasm is pretty much all water, and those guys are hydrophobic. So what about the second group there? These guys, these are called hydrophilic. Where do you think they're going to want to be? Outside. Beautiful. And then these guys are charged, and you can see that here we got a carboxyl group over here that's got an obvious negative charge. Got one over here. Those guys, if the pH is low enough, will pick up a proton, right? They've got a negative charge, but if the pH is low enough, there's going to be excess protons in the environment. They're going to grab one of those protons, so they're not any longer negatively charged. And the definition of acids are molecules that give off protons. So at a low pH, they're going to take them, but as the pH raises, then they're going to donate that proton. So we call them acidic amino acids. And these guys over here are basic because they do the opposite reaction. And you can see the positive charges on each one of those. It's going to be on these nitrogen groups that are right here on each one of those guys. All right. So we take amino acids, and here these might be the same amino acids, they might be different amino acids, but we see them here, and on this side we've got the carboxyl group with a negative charge. On this side of this one we've got the amino group with a positive charge. We can do that dehydration synthesis. We can kick out water, and we can form this covalent bond right here and do that dehydration synthesis to make that bond that's called a peptide bond. And so that's how proteins are formed. Ribosomes are constantly catalyzing this reaction and linking together amino acids to make peptides and then polypeptides. And then at one point, one or more polypeptide will fold together to make a functional protein, hopefully. And so you guys mentioned the primary and secondary sequence here just a little bit ago. Polypeptides can take on, they always have primary structure, but they may or may not have secondary, tertiary, and quaternary structure. So primary structure is simply the way those amino acids are linked together. So let's say the first one is methionine. You guys probably know that. Then we'll say the next one, I'm just going to use single letter abbreviations, alanine. Then we've got isoleucine, and you go on down the chain. Whatever that order is called the primary structure. So you change one amino acid in that long polypeptide chain, and the primary structure just changed. Everybody good with that? The side chains on each one of those amino acids we just looked at. They might be polar, they might be nonpolar, they might be positively charged, they might be negatively charged. They dictate what's going to happen as far as secondary structure goes. Those side chains and their nature. If they're charged, then they can play a role in forming hydrogen bonds and polar bonds, and we see that happening here with the side chains interacting between different amino acids. And if the polypeptide comes and folds this way, when scientists first saw that folded structure, they said, hey, it looks like the way we would fold our bed sheets when they come out of the laundry. They go up, they go down, they go up, they go down if you get a lot of those pleated sheets together, and so they call them beta pleated sheets. And then they saw a different type of structure, and here in the case, you get kind of helical structure form. You still get interactions happening between the side chains, but now those interactions, those amino acids that are interacting, tend to be closer together, and it causes the molecule to want to coil. So you get alpha helices in that case. That's secondary level structure. Let's say this is a pleated sheet, and this is an alpha helix over here. That's about as much as it can helix, and now they start to come together to fold as a molecule. Now we've got tertiary structure because we've got one type of secondary structure, this pleated sheet over here in this alpha helix, and the side chains on some of the other molecules are starting to interact together and draw those together to fold into a third structure, and that's called tertiary structure. That's the third level of structure. And then if you get multiple polypeptides coming together that are already formed, and now you've got two or more polypeptides coming together, that's called tertiary structure. Again, all of those are going to be held together by charge-charge interactions of the types of bonds that we just talked about. And those are all very important biologically. just to give you an example real quick, because I don't have many minutes, but when I was on the faculty at Southwestern Medical Center in the pathology department, my lab was looking at human diseases and genetic diseases that were caused by mutations in certain families. And we got cells, we got DNA, we got other things from those families, and I was interested in looking to see what's going on in their cells. They couldn't sit under these lights. I just told you that light's coming out. What I didn't tell you was that UV is also coming out. So you can thank the school while you're sitting here, you're actually getting a suntan. If you didn't know that, then now you know. UV is actually coming out of these lights as well. And these families that I was interested in have a genetic defect. We've got 5 minutes, so don't go away. I'm going to come back. They have a genetic defect that they can't fix, that the damage from the UV that's coming out of these lights causes to their skin. So if they sat under these lights for as long as you guys have in this one class, in about 30 minutes to an hour, they would have huge lesions all over their skin. Their skin would literally start dying and deteriorating because damage is being caused. Don't sue the school. Every light puts out UV, including the sun. And damage is being caused, but you all hopefully have healthy enzymes that can fix that damage before it actually kills the cells and causes those lesions on your skin. But these families didn't have. They somehow had defects, and nobody knew exactly what the defects were. So in my lab, we were looking at their cells and pulling out their chromosomes, and we sequenced through some of the genes that we knew that were involved in fixing damage from UV light, and we determined that the mutations were in one specific gene for these all 5 families that we were looking at, all genetically unrelated, but they had very similar disease state. And so pulled out the genes, sequenced those, and we found out the protein is pretty big. It's 454 amino acids long. Pretty big protein. And in one of the families, down at the end of the protein, I found there was one amino acid, just 3 amino acids from the end, and it was changed. There was a mutation that made the cell put a different amino acid in that position. It was a hydrophobic amino acid that was supposed to be there, and a second hydrophobic amino acid was put there in place of it. That's pretty conservative. But it didn't let the protein in those individuals fold correctly. The secondary structure came about, but when they started to interact together in the tertiary and the quaternary structure with the other polypeptides that are important in that process, they couldn't interact together because of that one amino acid out of 454 had changed because of a genetic mutation in those individuals. So that's just to give you an idea how important those side chains are in allowing those proteins to be able to fold correctly and function correctly down inside of your cell. So we get proteins with different domains, and domain 1 may have some function, and domain 2, 3 would have different functions, and we can actually now, we're getting pretty good information, and we can begin to look at the primary structure of genes, polypeptides, and figure out, hey, it must have this function based on that sequence. You already know that if you heat up proteins, you can denature proteins because you've done that in the frying pan. You took an egg, cracked it open, put it in the frying pan, added some heat. You saw the white of the egg, which was actually clear, go to white. As it was being heated up, all those proteins that are in the white of the egg were denaturing. They were unfolding, couldn't stay in solution anymore because they unfolded, and they denatured as you added heat. So that's what this is talking about. Those proteins had some function. Many of them were actually there with antimicrobial properties. You denatured them. They don't work anymore. And so heat can do that. There's a few other things, some chemicals that can denature proteins as well. So protein denaturation, pH, a few other solvents can break those bonds and cause them to unfold. That's what denaturing means in the case of proteins. And then they don't function anymore. Oh man, I don't want to let you go, but the clock says I really should. I guess I'll obey the clock.